Our protocol addresses an unmet need for a robust and reproducible method for quantitative analysis of joint changes in osteoarthritis development and progression. Our technique outlines specific and reproducible parameters for the quantitative analysis of changes in multiple joint tissues in osteoarthritis.
As this technique allows for quantitative analysis of osteoarthritis, it can be employed to compare the effects of specific treatments on multiple joint tissues in order to evaluate their therapeutic efficacy. Our protocol can also be applied to other osteoarthritis models or areas of musculoskeletal research in which quantification of cartilage and bone is needed.
Although the system is straightforward and intuitive, training sessions are still necessary to practice obtaining measurements in a repeated manner to achieve reproducibility.
Vengadeshprabhu Karuppagounder, a postdoctoral scholar from our laboratory, will now be demonstrating our procedure.
Begin by turning on the microscope, camera, computer, and touchscreen monitor. Select the 4x objective and click the software icon to launch the software program. Place the slide on the microscope stage and center the sample within the measurement window, ensuring that the measurement grid is set to the proper magnification to match the objective and that both the tibial and femoral articular surface and tibial subchondral bone are included within the image regions of interest. Then, under the File menu, select Read Settings. In the Settings window, select the appropriate settings file for the parameters to be measured.